So first off, for this week, I've pulled out several amino acids with clinical relevance, either in the context of newborn screening or deficiencies. So we're not going to talk about or even begin to focus on the metabolism of all 20 amino acids. Rather, we'll focus on very small subsets. So we'll start with the metabolism of aromatic amino acids. Before we focus on any specific amino acid, I want to talk about a specific cofactor we're going to need, and that's tetrahydrobiopterin, or BH4. So this cofactor is required for the metabolism of phenylalanine, tyrosine, and tryptophan, all three amino acids we're going to talk about. It's synthesized from GTP, and again is required for hydroxylation reactions, and most notably, that's the conversion of phenylalanine to tyrosine. What's also important to note is that deficiencies in the cofactor BH4 can present in a similar fashion to deficiencies in our enzyme for phenylalanine metabolism. So if we start with the metabolism of phenylalanine, it's important to note that phenylalanine is an essential amino acid, and hydroxylation of phenylalanine by the enzyme phenylalanine hydroxylase results in the production of tyrosine. Again, the conversion here from phenylalanine to tyrosine requires our cofactor BH4, and deficiencies in either the enzyme phenylalanine hydroxylase or the cofactor BH4 can result in the clinical presentation of phenylketonuria. In the clinical conditions when metabolism of phenylalanine is impaired, this also can result in tyrosine becoming a conditionally essential amino acid, meaning it must be supplied from an exogenous source. You'll also note that phenylalanine can also undergo a transamination reaction using a transaminase and the cofactor peroxidyl phosphate, and that results in the generation of phenylacetate or phenylactate. So the tyrosine produced from the metabolism of phenylalanine is required for the production of melanin. So the important enzyme here is tyrosinase, and deficiencies here can cause disorders such as hypopigmentation disorder due to decreased production of melanin. Tyrosine is also required as a substrate for thyroid hormone synthesis. So as an alternative, tyrosine can be transaminated using a transaminase to generate intermediates that can enter into the TCA cycle, both fumarate and acetoacetate. And deficiencies in the metabolism of this keto acid often result here at our enzyme homogentisate oxidase. So deficiencies in the enzyme here can result in the elevation of homogentisate. This metabolite is spontaneously converted to alkapton and results in the presentation referred to as alkaptonuria. Our final amino acid to talk about is tryptophan. So tryptophan is also an essential amino acid that's both ketogenic and glucogenic, as it can be oxidized to produce alanine or acetyl-CoA. The ring structure can also be used to synthesize niacin. So the initial metabolism of tryptophan requires BH4 as a cofactor, and the downstream products are serotonin and melatonin. So deficiencies in the metabolism of tryptophan will result in deficiencies of both of these important products. So let's finish by looking at a clinical question. So here we have a 52-year-old male who sought medical help complaining of progressive hyperchromic, hyperpigmented areas on a second finger. He's also noted darkening urine. So a biopsy and urine sample tested positive for homogentisic acid. And based on these findings, a diagnosis of alkaptonuria is made. So I'm asking you the question, this diagnosis is consistent with a deficiency of which of the following enzymes? So to unravel this question, we don't need to know much, if anything, about the clinical presentation. What we need to know is what amino acid is metabolized to homogentisic acid. So first off, phenylalanine hydroxylase is required in the metabolism of phenylalanine to tyrosine. So this is not a great choice here. Second, alpha-keto acid dehydrogenase we'll see later is responsible for branched-chain amino acid metabolism, so we can eliminate this off the bat simply because we didn't talk about it here. So tyrosinase is a deficiency that results in reduced melanin production. So this would result in hypopigmentation, not hyperpigmentation. So again, we can cross this enzyme out, leaving us with homogentisate oxidase, which is in fact our correct answer. If you remember from previous slides, this is responsible for the metabolism of the keto acid of tyrosine into TCA cycle intermediates. And deficiencies at this point in the pathway result in an elevation of homogentisic acid. So focus on the few key enzymes we've talked about here, and we'll move on to talk about a few other amino acids in the next discussions.